Okay, let's start off the class today. We are going to start with the questions. Some students, not some students, only one student, by the way. They put the last week. Okay, so yeah, let me start with this sentence. So can you hear me? So if you can hear me, you can leave the chat or raise the hand, because I should know the my microphone is working. Okay, so this is working. So that's good. Okay, so let's start with the questions from Minza last Wednesday. So she put 5 questions. So first and second is quite similar. So the question was, what is the delta on Fisher's single locus model and how to calculate those delta in the experiment. So the delta defined by Ronald Fisher at the time was the arbitrary one. So when, I mean, the delta is the situation whether we set the assumption that where the locus follows additive or dominance, right? So he put some different situations, like, you know, different categories, you can say that. So for instance, the locus or allele follow the additive situation, then we have to see sort of the no phenotypic changes in a heterozygote. But if the heterozygotes are biased towards certain direction, so that case, either one, you know, the phenotype by Mendel or the other phenotype, again Mendel, then we can say that this is the dominant situation, right? So delta is the situation where we describe the whether the locus follow that model or not. Okay, and then there must be a way. I mean, the students assume that there must be a way to define delta in the situation, but the issue is that there is no such an allele that we can say that it is obviously and 100% affects phenotype, right? So as now we know that and also as Fisher indicate that most of the traits and disease are actually following the polygenic situation. So I mean, we could only you know, give examples of the Mendelian situation, but most of the situations are actually following the polygenic situation. So if you want to measure that delta value on the situation, that case you have to know the you have to know that the locus only single locus affect on the phenotype, and also there is a very objective way to measure phenotype. But I mean, we know that in the realistic world there is no such things exist in our field. Okay, and the third question is, if you want to know about the genes related to height, then her question is that whether we have to recruit the people, the samples with the same conditions about the height, and also we have to exclude all possible the variables affect on height. But you know, this is again, it is also an unrealistic situation, and so I mean, when you calculate when you study the genes related to height, or I mean, more technically, we say that the genes associated with height, that case we normally recruit the almost randomized samples. Then how we can calculate the genes related to the height, that case we use the sufficient number of samples. So we obtained enough power for study height genes. That case we can calculate we can identify the genes related to height. Okay, that was the question. And then I'm not sure whether this question is about the genes or alleles, but calculate genes is more sophisticated and more complex way. So we normally calculate the alleles first. So alleles in the modern context, it has to be mutation or variant, genetic variant. So the case, I mean, as I mentioned, that we used almost the randomly sample individuals from the population. Then we compare the heights across the individuals and calculate the alleles associated with height. Then once we define alleles, then we think about the genes related to height. That was sort of the flow. And then and we're going to discuss this issue more properly on the session 5. And the fourth questions from Minsa was, once we know about the allele related to height or about height, then her question is whether, I mean, if the allele is homozygous, then she thought that there must be huge impact on the height. That could be yes or no. I mean, the reason why I hesitate to answer this question, because you have to know about the different scenarios of additive and dominant alleles on the height, and that will be discussed in the session 2 and 4 and session 2 and 5, which is the genes and heritability. Then we're going to discuss about the additive factors and dominance factors. I mean, if you're not really patient enough, then you can go to slide and check it. But the short answer is that the homozygous or heterozygous in this case is not relevant to how much trait value you're going to have in the population. That is totally incorrect understanding about the heights. We are quite different from the trait Mendel look at the time, and knowing the situation is the critical things why we learning the polygenic model. Okay, and the fifth question was the locus is the physical position on chromosome. That was the answer in other questions at the time. And then Fisher's single locus model used the locus, and our question was that the locus in the term is also the physical position. There was no, because at the time Fisher didn't know anything about chromosome and also didn't know anything about the meaning of the chromosomes, right? So meaning of the chromosomes means that the chromosome is the physical locations of the alleles and genes, and the initial understanding actually from the person Thomas Morgan in 1918 and also between 1920 and 30. So probably the Morgan was the first person to appreciate the situation, and Ronald Fisher at the time, I mean the 1910s, he didn't know anything about the position. So that case, single locus model, in this context, locus is just merely equal meaning as allele, and that was the heredity factor defined by Mendel. That was quite similar concept. Okay, so and the last question from Minsa was that I mean, can we use the locus as just the meaning of allele? Yes, yeah, that's fine. Okay, so that was the question, and if you have more questions, then leave yours on the Slack, and then adjust your understanding and thought and developing your ideas onto the genetic terms and contents. Otherwise, it will be difficult when you prepare the exams. Okay, so back to the syllabus. We are on the session 2 and 3, which is the Hardy-Weinberg equilibrium. We're going to learn these things on today. And then the Wednesday, we're going to follow up with the chromosome and sex. There was the about the Thomas Morgan, and this is the forerunner of the concept of gene. Chromosome and sex is about the meiosis and then the Mendelian factors, and then followed by the Thomas Morgan's idea about the gene and the heritability. So this week we're going to cover these two, and next week we're going to cover these two. Then the week after the Monday is the review session. Then Wednesday we're going to have exam. So think about the timeline and prepare your exam. Otherwise, one thing I should tell you is that I'm not taking questions just 5 hours before the exams or 10 hours before the exams. It happens every semester. So if you have questions, if you have any comments about the exams, please use the time this week and next week, right? I'm going to do the review session Q&A on the Monday, just 2 days before the exam, but that time you have to be prepared about your exams. Otherwise, I mean, there's no way we can do Q&A session, because I'm not 100% doing class on my time, because I'm doing other things. I'm doing research. I'm doing other whatever the politics, admin stuff. So I have no physical time to do that. So I mean, some students, I mean, think that, you know, I can prepare this, you know, easy Mendelian genetics just 2 or 3 days before the exam. I mean, don't do that stupid things. I mean, there's no such things can work in the exam. I can say that. So, so I should tell you that you have to, at least you have to give me some questions from this week. I mean, if you have no questions about the, you know, the slides or the class, I mean, there must be something wrong, right? I mean, so please be on that issue. Okay, so Hardy-Weinberg equilibrium. So we're going to learn about the 4 stuffs about in this class. So first one is the very basic introduction of allele and allele frequency, and we're going to introduce the concept of the Hardy-Weinberg equilibrium, and then examples and how to calculate the Hardy-Weinberg equilibrium. And you can have some textbook from this book, the chapter 14, and also you can access to the textbook freely on our university library. And also some of the learning materials is beyond the paywall, so you have to use our library internet or use the Sci-Hub for your access. Okay, so allele. So, I mean, as we know that the allele is the basic unit for the genotype and also the phenotype, right? So, and also allele as the unit, the Mendel defined at the time, called the hereditary factors, right? So allele is the term describing that the Mendelian factors. And as we know that the alleles constitute the phenotype, and the phenotype is quite different by individuals in the population. So allele has to exist as a with a different frequency in a population. So in other words, we can say that the proportion of the chromosomes in a population carrying the allele of the traits or disease, right? So when we look at the populations, when we look at the certain phenotypes like eye colors, some people has the black, brown, or blue, and those eye colors has to be different proportions in the population, and also this different proportional population followed by the proportion of the alleles in the chromosomes. And also, I mean, other than eye colors, we can also look at the Bartlett's patterns or type 2 diabetes as well. So first thing you have to know about is the allele proportion or allele frequency is quite equivalent term to the phenotypic distribution in a population. So that is the one thing. And the other one is how we define phenotypes, right? So as I mentioned, the eye colors, we can say that this is the whatever, the blue eye or brown eye or black eye, but some eyes could be like a bluish, some eyes close to, you know, brownish. So this is the situation we can define the phenotypes a bit differently. And in other words, there must be a way we have to measure this phenotype objectively, right? So, and this is the why wearable devices or other, you know, additional machines is critical and also important, because this is the framework we measure the phenotype objective way, right? Okay, and also the, like, you know, baldness pattern. So this is quite, you know, fun cartoons, but this is actually the how we define baldness in a population, right? So if you look, this is the definition from UK Biobank data, and UK Biobank data is the population level studies from British Society, and they're looking at the different people and different males, different females, and they define how males or females has the baldness in their population, right? And as you know, that the baldness is also very various format in a population, but for the simplicity, they define the baldness into 4 categories, right? So pattern 1 is no baldness, so no hair loss, and pattern 2 has, like, you know, some M shape, and pattern 3 has the O shape, and pattern 4 has the miserable. So this is the way how we define the phenotype in a population or in a clinic situation. So defining phenotype is, I mean, looks simple, but it's quite sophisticated, and also it's quite something we have to study heavily, right? And otherwise, the genetic underpinning is quite different between the phenotypes. So we have to measure the phenotype exactly, and also think about the, you know, whatever the genetic studies. And this is the same situation for type 2 diabetes or other diseases as well. Okay, so as we learned from Fisher's model, disease or traits are determined by different combinations of alleles and also environmental factors. But for the simplicity, genetic factors is much easier way to, you know, put it into the formula and also calculate the contribution. So we at the first year we studied the genetic factors underlying that phenotypes. Okay, and then allele frequency is equal meaning as the proportion of the alleles in the chromosome in population. For the simplicity, we called it allele frequency, and this indicate that the probability of observed genotypes in a given population. So when you look at certain phenotype, and that phenotype is developed by certain allele, and that allele has the certain frequency in a population, and the frequency is really matter, because this is the criteria whether we, you know, we can think about this disease seriously or not, right? So allele frequency is the key value we have to think about in our genetic study. Okay, so let's look at the examples of one allele, right? Allele is, as I mentioned, that this is the key term in Mendelian genetics, but in modern sense, I mean, we still use allele, but we more frequently use different words, which is the variant, right, or mutation. But I mean, I'll, you know, discuss later. The mutation and variance is slightly different, and variance is more precise and more comprehensive words, and also many various types in the variants. So this is the SNV, which is the single nucleotide variant, and we have the database of these variants. So we have the, you know, list of the single nucleotide variants, or we have the indel variants, we have the copy number variants, different types of the variants, and those variants are equal to allele in the context of modern genetics. Okay, so let's look at this database, and let's go to this link. So if you see the bluish character, that will be hyperlink, and you can click that, then you can go to the database called dbSNP. This is the database of the SNP. Very brilliant naming. And this is the ID number, which is the RS75 something something. Then which organisms this allele or SNPs from. This is from the Homo sapiens, which is you and your friends, not me. And the position on the chromosome 1 and this position. So this is the physical position. And you can click that one in here. Then you see where that is located, right? So this is your DNA sequence and where these the SNPs or variants are located, right? This is quite interesting stuff because we learn about Mendelian genetics, like Mendelian notation. For instance, like large R and small r is heterozygous, or large R and large R is the homozygous, whatever, or AB, AA, or BB, something. That is the notation invented by Mendel in 1886. But the notation is still existing in a university textbook and still, taught for, the biology students, but we don't use anymore. So Mendelian notation has to be converted to the modern sense, which is the DNA sequence. But, this is quite, leap between your, this is quite, leap of your understanding between Mendelian genetics and DNA sequence, because, we don't know anything about, DNA sequence, how to, identify this sequence or blah, blah, blah. But one thing you have to know about that is, keep in mind that the Mendelian notation, like, heterozygous, whatever the homozygous, is actually has to convert it to the DNA sequence, or the changes on the DNA sequence. That is the equal to the concept of allele in the modern sense. And we're going to discuss this, identification in session 4 and 5. Okay, so back to here. So we have the additional information about the frequency. So this is the frequency information about this allele. Okay, so this allele is G to A, right? So the G is the one has the ancestral DNA sequence. So somewhere in the old humans has G, but the modern humans or some humans has C, right? So that case, the allele, right? So G is changed to A, right? So then we can say that the homozygous, according to the Mendelian genotype, that case, the homozygous reference has to be GG, right? And the heterozygous is GA, right? And then the recessive, or recessive is wrong. The homozygous variants is the AA, right? Because we don't know anything about the dominance on this allele, right? So 3 genotypes is possible, which is GG, GA, and AA, right? So we want to know about the frequency of how many people has A in our population, right? So this is the frequency reports. So there are the tens of the reports about the frequency. So according to TopMed, the database, data source, they look at the almost 120,000 alleles, and out of that 30–60% has the A allele, right? So rest, the 70% has the G allele. But things you have to know about that is these frequency values is slightly differ by the project, right? So TopMed is from the United States, Nomads from the United States as well, 1000 Genome from the international project, Korean is obviously from Korean Genome, and Twins UK from UK, and Korean 1K is obviously from Korea, and GeoNL is from the Netherlands, and Qatari from Qatar, obviously, and Vietnamese, obviously from Vietnamese. So each ancestry, each nation, they have different composition of the population, ancestry population. So the alleles is slightly different distribution across the population. And also the calculation of the allele frequency is differed by the number of participants used in the study too, right? So that is the one thing. So this is why the frequency is slightly different from the studies. And also we have to keep in mind that if you want to study Koreans, then you have to matching the numbers with the Korean statistics, right? Okay, so this is the ID number, and this is the whatever the position, allele, and the frequency is the 36. Yep. And the population means the TOMED studies and how many people sampled from here. But what I explained is it's actually, I think it's actually wrong. The reason is that give me a sec. Okay, so I actually put these numbers, these samples recruited in the TOMED studies, but this is actually the wrong number. The reason is that the humans has the diploid genome, which is that we have the 2 chromosomes. We're going to learn in the next session. And that number has to be reduced by half because the allele has to be calculated on the 2 strands. So that is easy calculation. Okay, let me fix that number. This is actually 62784 because it's really 5 by 6, 8 divided by 2. Because this 2 is from the diploid genome, right? So it has to be divided by half. Okay, so yeah, please bear with me. Sometimes the typos error. Okay, I think that's all, and we're going to come up with the other, definitions like gene consequence or something in a later section. One for the curiosity. So if you look at the dbSNP database, you can also click the clinical significance. Then if this allele is associated with certain disease, then it comes in this session, right? And the frequency and other informations as well, right? Okay, cool. Moving on to the next one. Okay, so allele transmission to the next generation. So allele transmission is obviously follow the Mendelian first law. So offspring inherit one allele from each parent, right? And parents transmit allele independently of each other. So what that means is the father's transmission of one allele and mother's transmission of one allele is totally independent to each other. There is no way we can interact between these two processes, right? So this is totally randomized and also independent process. And within a parent, transmission is random, as I mentioned that, and with the equal probability for each allele, right? So if you think about the meiosis, then meiosis give you the 2 gametes from one cell, one germ cell, and that one has the random segregation between the Mendelian factors. Okay, and this is the totally independent process, and no matter what you have non-genetic forces, like, whatever environmental factors, smokings or the air pollution or aliens attack, it's all independent process. This Mendelian laws is always happen independently between parents. Okay, so that is important assumption for understanding the transmission. So let's put it into the probability formula. So probability a parent passes on a allele is the 0.5 because this is the 50% probability. So this is quite simple things, but this is quite important because this 50% probability never change it because it's fixed in our nature, right? So parents has AA allele, both large A, then gametes has the A and A, and the parents has the AA heterozygotes, then gametes has the whatever the large A allele or small A allele, and that one is the 50% chance transmit to the offspring, right? So there is no way we can invert that situation. Okay, so if you think about that randomized the probability, then we can easily calculate how the probability of the genotypes you will have in the next generation. So let's say if you have the frequency of allele in a population, this is not the probability of the transmission. This is the frequency of the allele in a population. So large A allele is the 70% and small A allele is the 30%. Then you can easily calculate that in the next generation, the probability of the homozygous large A is the 0.49, right? Because this is the 0.7 times 0.7. So 7 times 7 is 49. And the heterozygous probability is this situation is 0.21. So, but that case you have the 2. So the probability is double than that. Then the recessive is the 0.09, right? So we can, once you know the frequency of the allele, then you can easily calculate the frequency of the genotypes in the next generation. So that is the easiest way, and we call this one is the Punnett square. And this is, one of the easy things we can think about allele composition. Okay, so let's put it into different the discussion and also the major topic in this section, which is the Hardy-Weinberg equilibrium. So Weinberg is the German, so it has to be V sounds on W, so Hardy-Weinberg. And the Hardy-Weinberg equilibrium is the critical thing. You have to understand the concept of the Mendelian factors in early genetics. So the questions we have in this topic is why the dominant characters did not become more frequent from generation to generation. So the question is the dominant allele has to be appeared in the heterozygous. So after many generations of the human history, this dominant allele has to have the dominant frequency in a population, but it isn't, right? So that question is actually rising in a Punnett's lecture in 1908 and raised by Yule. So you raised the question, as I mentioned in the previous, during the Punnett's lecture on the Mendelism in relation to disease. So Yule's question was, if in a population, brachydactyly, which is the short finger, so this is the disease has the short finger, and the people know this disease is the dominant pattern. So, I mean, he said that this disease was determined by dominant gene because they know about this disease from the pedigree study, and they know that this disease is follow the dominant patterns, and this disease is follow the dominant patterns and do the random mating. The expectation is we have about 3-to-1 ratio of this disease in our population as Mendel's ratio, right? But your question was that we see most people do not have this disease. Most people has the normal finger, not the short finger. So what is the reason for that? So is that the mean that the Mendelian genetics is wrong, right? So Punnett got the question, and Punnett thought that this is not true, but he couldn't prove it, right? So that was the question he didn't resolve at the time. Then Punnett brought this question to Hardy, which is the clinical doctor and also the mathematician, at one high table dinner. High table dinner is like a social club, and they talking about whatever the upper class discussion. And Punnett brought the question, and Hardy gave a solution by using a frequency of 2 alleles easily using the equation. And then he, published their work in later. But anyway, so Hardy's conclusion was that segregation in Mendelian inheritance law is not affected by segregant, right? So segregant is the allele, right? So segregation and segregant is independent entity, right? And then Hardy called this situation is stable, right? Okay, so let's put on this Hardy's calculation. So let's assume the few situations. So once we simplify this analysis, Hardy imagined the situation that where large population choice of the sexual partner would be random. So there is no preference, there is no associative way, it's randomly happening. So your mating is randomly happening in the large population. Otherwise, then if there is a the preference on the mating, then there must be a distortion in a frequency. So the mating is random, and it has to be in the large population. And then other assumption is that at the single locus, there should be only two factors, which is the allele. Some case a locus has the three factors, 3 alleles, or 4 alleles, or 10 alleles. So it has to be only 2 alleles at the locus, right? So for the generation n, let Pn be the frequency of the genotype large A and large A. And then the 2qn, that of the heterozygous, and Rn of the small a and small a, which is the homozygous small a, right? So of course, these three different scenario has to come up with the sum up with the 1, because this is the entire composition of the population, right? And then Hardy also assumed that none of these genotype leads to the excess of mortality or the decrease in fertility when compared to other genotypes. Otherwise, it has to be distorted or it has to be disappeared during the multiple generations, right? So this is all clear. Okay, then the frequency in the next generation, which is the n+1, can be easily computed by the one randomly chosen individuals in a generation n, and then transmit allele large A with a small a probability Pn plus Qn, right? So, let's put the simplest way. So genotype is the homozygous large A, which is the Pn. And then allele large A is transmit with the 100% chance, right? So we can easily say Pn. Then the heterozygous situation, the probability of having large A is the 50% chance, because you're going to transmit either large A or small a, right? So you can also calculate the small a, the probability of the transmission in this situation, Qn plus Rn. So if you put this in each situation in a Punnett square, then you can easily calculate that in the next generation, large A and large A is the probability of Pn plus Qn times squares, right? And the heterozygous situation is the Pn plus Qn and the Q plus Rn, because this is the probability of here, right? Then the homozygous of small a is also the same situations too. Okay, then we can easily put the probability of 3 different genotypes into the Hardy's first assumption, which is the Pn plus 1, Q 2Qn plus 1, Rn plus 1. Then what Hardy found that the Pn plus 1 is equal to Pn plus Qn times 2, because this is from here, right? And this heterozygous also from this situation, and the recessive situation is like the dominant homozygous situations too. So then once you summarize the probability in the next generation, then you can, as you can see here, this is exactly following the same formula as you define in a initial generation. So in other words, we can say that this probability is constant and stable during the generation, and this is exactly same probability as the first generation, right? Okay, so when we start the initial definitions of the probability, we start off the entire population is sum up to 1, which is the P plus 2Q plus R equal 1. So we can see that all of the equations about the heterozygous and homozygous, whatever the dominant or recessive, is actually ends up with the same condition, which is the Q squares equal to PR, right? So let's derive from the equation 1. So equation 1 gives the P equal P plus Q times squares. This is the equal to P2 plus 2PQ plus Q2, which is the equivalent to the P bracket and 1 minus P minus 2Q equal to Q2, right? And finally, it's the same as the PR equal Q2, right? So this is the easy formula given your high school things, and this is the same derivation from your equation 2. Okay, so let's put the initial generation arbitrarily. So let's put the P0, Q0, and R0, and that is equal to, as we define in the sums of the genotypes in the population. Then the Hardy noticed that Q2 in the next generation is actually equal to P1 and R1 times, right? So this is the same situation you derive from the equation 1, right? So in other words, and as we see in the previous slides, these 3 genotypes is stable and also it follows the under the equilibrium situation. Then this is the as we mentioned that whatever the generation on the 3 genotypes is actually equal to the initial generation or the second generation, and it never going to be of the above that the sums, which is the 1, and whatever the generation changes and generation grows, and it has the same the proportion in the population, right? Okay. And then we can put this equation in more simplistic version, which is that the x equal P0 plus Q0, which is the frequency of allele large A in a generation 0. Then this is equal to 1 minus x equal Q0 plus R0, right? So this is the frequency of the small allele of the small a, right? So because it has complementary to each other, so the sum of it has to be come up with the equal to 1. So we use that simple notation and put it into the equations we see in a previous slide 1 to 3. Then we can easily derive that the situation, which is the Pn equal to x squared, and 2qn is 2x times 1 minus x, and Rn is the 1 minus x is 2, right? And again, if you plot that one into the graph, then it perfectly has the exact matching the balance, whatever the frequency of the large A allele or small A allele, whatever changing that one is constantly sum up to the 1, because that is the total proportion in a population, and that one is exactly complementary to each other. Then we can say that situation is the stable or under equilibrium, right? So we can easily demonstrate that the 3 possible genotypes is always following the same proportion in a population, and we can easily derive from the other populations. Okay, so before this Hardy-Weinberg law, calculations of the effect of natural selection required one to keep track of 3 variables, which is the 3 genotype frequencies, right? So before the Hardy-Weinberg, we have to know about the frequency of the 3 genotypes from the certain phenotype, right? So think about the Mendelian things, right? So always your calculation or algebra required to do more than 2 situations, which is the 3 genotypes, and put it into the 3 variables. So we can say that this is quite complicated, but here, now we know that the allele frequency is interchangeable with the genotype frequency because as we derived from this slide, right? And this allele frequency on a single locus, which is the binary one, that one is also complementary to each other and sum up to 1 in a population. Then that situation, once you know the one frequency, then you can calculate the other alleles frequency in our population, right? So let's assume that we have the 2 alleles like a P and Q, then you can easily put it into equation, which is P plus Q times squared, and that is the P squared plus 2PQ plus Q2 equal 1, and that is exactly same as the P plus Q1 because you can put the square root, then you can rip off this P plus Q into the 1, right? This is the simple mathematics, think about your high school. Okay, so the assumption, as I mentioned, that the once you know the one allele frequency, then you can easily trace back the other allele frequency in our population. That is that is okay, and that is easy and simple things. And but the question is why these simple things is really matter in our biology, in our time, right? I mean, even though it looks quite simple, but at the time, as I mentioned, the biology is less prone to formulate something, right? So there was quite some meaning in biology because I mean we formulate something and then we use the simple formula to describe the biological phenomena, right? So there was one fundamental, the idea is about how we describe the alleles, then it followed with the idea of the genetic linkage by Thomas Morgan, and people start think about, the physical position and phenotypic, the interaction, and then people think about the central dogma, blah, blah, blah. So put it into the, mathematical model, whatever, it's a simple thing, it's critical start for the, the downstream study. Okay, and then, when we think about the Hardy-Weinberg equilibrium, we assume lots of things, right? So first one is the population has to have random mating, right? And no differential fertility of the genotypes, that is the key thing. And also, the reason is the natural selection is not acting on a certain locus in questions. And the third thing is equal genotype frequency in 2 sexes, right? So, I mean, if that is put it on the sex chromosome, then we kind of easily calculate that the Hardy-Weinberg situation. And also neither mutation or migration should happen, and no differential variability and infinite population size. That is sort of all assumption the Hardy-Weinberg equilibrium is working in our genome. But in the real world, this Hardy-Weinberg equilibrium is not always working because the old assumption is not realistic, and each individuals and also each population has slightly different situation not following that assumption. So studying the population characteristic, as to this assumption is the field of population genetics and major topics we want to study in a clinical field and also the nationwide genome project as well. Okay, so once you know about the Hardy-Weinberg, and before we moving on to that, a few things we have to know about the implication. So first one is the Mendelian segregation is preserved in any organism with the sexual reproduction, regardless of allele frequency in the population. Preserved is something. So I mean that means they never change it in our nature, right? Whatever the environmental factors. So any organisms with the sexual reproduction, it always have the Mendelian segregation pattern, right? And in this, under the situation, Hardy-Weinberg principles as the simple formula representing the Mendelian segregation. So this is something, and calculate the deviation of the Hardy-Weinberg equilibrium is the way we look at the population structure. So we're going to discuss this in the session 3. Okay, so a few more implications on this. So, I mean, as I mentioned in the previous slide, under the Hardy-Weinberg assumption, the frequency of allele is unchanged over generation, right? So Mendelian segregation is all working and randomized and equal probability, which is a 50% chance. And the allele frequency in a natural population in real world could be altered over the generations due to the non-Mendelian effect, or we can say the population effect. And this is the critical, theme in a modern genetics and also the whatever biomedical genetics. So I put this issue in our session 3. We're going to learn this issue in our next session. And then this is the whatever. If I have to, pick up one key sentence in this time, I have to keep this one for you, because the dominance in the genetics, dominance does not mean frequency. It's a molecular phenotype. If you understand this sentence properly, then you're done. This is critical. So dominance, somehow, if you translate in Korean, the dominance means that, this is like, something indicating the major, majority of within the population. But dominance in Mendelian genetics and dominance in genetics, this is the term indicating the molecular phenotype. And molecular phenotype is determined on a DNA sequence, and how DNA sequence changes can be represented in your phenotype. So understanding that flow is the key things in genetics and also in molecular biology, right? So the question is that how, if the allele frequency is not changed over the generation, without a population effect, and how an allele frequency has to be set to in an initial generation. That is the question for you. So think about that question in your mind, because the allele has to arise in sometime in our generation, and that is transmit to the next generation, and that the alleles constitute some proportion in our population. But then the question is how allele arising at the initial generation, right? Because this is the missing link between the allele frequency not changed according to Hardy-Weinberg, but then how to set that initial frequency in a population, and what is that? So the question is critical, because if you keep this question in your mind, then you can enjoy the session on the session 3, because this is the all the following notion from Hardy-Weinberg to the population genetics, right? And this question is then related to the topics in the biomedical genetics, right? So as we discussed in the last Friday lecture in the introductory department course, I mentioned about the biomedical genetics and the questions from the clinical doctors, and what they failed understanding modern genetics is to think about how allele frequency has the meaning, has the implication in their clinical research, right? Unfortunately, most of the clinical researchers in this country have no time to study the modern genetics, because they are memorize the things and they memorize the technical things. They're technicians, they're not researchers. Most of them are not researchers. So thinking about the latest sense of the genetics into their field is something insufficient, right? So, but they're thinking about the sense of the allele frequency and also the meaning of the allele in a population context is the key things, because we see more and more evidence on a population genetics in a disease study, and how to interpret the genes or alleles associated with certain disease is the critical, right? And that one has to be come up with the population interpretation. Okay, so this is the question for you for the later section, and also we're going to discuss this in the session 3. Okay, so let's back to the allele frequency and Hardy-Weinberg, right? So first example is the hair color. So let's assume that we have the 3 hair colors in our population, and then we know only 2 things. So first one is the frequency of the blue hair, which is the 9% in our population, and the hair color determined by the 3 genotypes and 2 alleles, which is the blue small h, and the black small h and large h, and purple, which is the large h homozygous. And then we can calculate the frequency of black and purple hair according to Hardy-Weinberg equilibrium, and that is quite, the easy things, right? Because I mean, this is the easy mathematical questions, right? And the example 2, this is more recent evidence on the CRISPR topic. So gene CCLR5. This is the gene encoding the protein, which is on the cell surface, cytokine receptor. Then that is the acting on the entry point for the certain traits of the HIV in the AIDS, right? And some scientists in China, he claimed that if you use the CRISPR-Cas9 system and delete the gene, then the baby born in a generation should have the proper immune system to protect AIDS, protect from AIDS, right? And this guy, I don't remember his name, but he did the CRISPR-Cas9 experiment and really delete the genes in embryo, and the 2 babies born in, I think it's 2018 or 2019, sometime there. So individuals are homozygous for this gene allele. Whatever, yeah, that the idea, and then we can calculate, how many people has that issue in the population. This is the same, calculation as we learned from the Hardy-Weinberg. The why I put this one, but anyway, yeah, there is some repute for this study about his calculation was wrong, and some of the biological questions was totally failed. And I don't know. I heard that this guy is disappeared by the government, but anyway, yeah, that was the wrong question, and that was the wrong assumption on the science, and yeah, and then. Yeah, okay, so that one is also affecting another phenotype. So that was the issue. Okay. Okay, and the third example is the Phenylketonuria disease. This is the congenital disease with the metabolic issue, and that is following the recessive pattern. So we call it recessive disorder. So frequency of this PKU is approximately 1 in 40,000 people in our country, right? And then we can calculate that what are the chances of Korean couple without known family history of PKU having child with PKU, because this PKU follow the recessive disorder. So you and your partner has the heterozygous genotype, then in case your child could have the 25% of having PKU in his phenotype, right? So you can easily calculate, followed by the Hardy-Weinberg principles as well, right? So first thing, we can calculate from the frequency of the affected individuals by PKU. That is 1 in 40,000. That is equal to Q2, which is the recessive pattern and the recessive genotype. So you can easily calculate the allele frequency of Q, right? So this is the square root, and you can calculate this is the 0.005, right? And the P allele frequency is 1 minus 0.005, which is 0.995, right? So you can easily calculate the frequency of heterozygous genotype, which is the 2PQ, and this is equal to 0.01. and this is approximately 1%. So this is the frequency of heterozygous carrier. Carrier means that the person has the heterozygous genotypes but do not have disease, right? So the carrier frequency in the Korean population is the 1%, right? So frequency of having child with PKU when both parents are carriers, right? In that case, you have the 25% of having PKU child because this is the recessive patterns, right? So think about the situation, large AA and 2 heterozygous and 1 small AA, right? So that is the 1-to-1 ratio. So that is the 25%. So the chance that the couple's child with PKU is the 0.01 times 25%, 0.25. So that is approximately 0.00025, which is 1 in 40,000 chances having the PKU. So we can easily calculate that the chance from the Hardy-Weinberg principles. But don't be fooled yourself that this is the same number, but this is the different, the answers, right? So I mean, coincidence. This is the coincidence, or accidentally, that has the same numbers. But actually, if you calculate in these different examples, it always different, the numbers. So don't be fooled yourself. Yeah. Okay. So this is the way we calculate the frequency of allele according to the Hardy-Weinberg principles. Okay, so more proper way we can calculate that Hardy-Weinberg situation, which is using the chi-square test. Okay, testing Hardy-Weinberg equilibrium. So when locus is not in the Hardy-Weinberg equilibrium, then this suggests that one or more of the Hardy-Weinberg assumption is false. That is obvious, right? And departure from Hardy-Weinberg equilibrium or deviation from Hardy-Weinberg equilibrium, in other words, Hardy-Weinberg disequilibrium, has been used to infer the existence of natural selection, because this is the, one of our assumptions: no natural selection, random mating, no fertility reduced. So, if that is, not Hardy-Weinberg equilibrium, so we can easily think about that there must be existence of natural selection. And then we can also argue that the existence of non-random mating, right? So some animals or humans, we don't do random mating. We don't, we want to marry with, we really want to marry, right? So non-random mating. And also sometimes, the departure means that the genotyping errors, right? Because we measure something, the measurement itself is erroneous, right? So Hardy-Weinberg equilibrium indicating that genotyping errors, right? So finding a factor causing this deviation from Hardy-Weinberg equilibrium is critical to understand biological phenomena in your population, or also improve our analysis too. Okay, so that case, we can use 2 different types of tests. One is the chi-square test, and the second is the exact test. So if you learn basic statistics, you know that these two are quite similar, but in this lecture, we only introduced the chi-square test. And that is initially introduced by American naturalist Harris in 1920s. And he first used the Pearson statistics at the time, but then people developed that idea and used the chi-square or exact test, blah, blah, blah. Okay, so what chi-square statistics assume in a Hardy-Weinberg equilibrium? So chi-test allow you to use statistics to determine whether locus is of your interest is under Hardy-Weinberg equilibrium. So the null hypothesis in this situation is assumed that there is no substantial statistical deviation between the observed values and expected values, right? So this is the typical null hypothesis. And then you measure the chi statistics using the observed value in your experiments and the expected value according to Mendelian ratio, right? So what that means is that the locus has to follow the Mendelian factors, because the Mendelian ratio, because that is the natural situation has to happen in our population, right? So your null hypothesis has to be the expected values with the Mendelian ratio, right? Then you describe your observation. So that means the observed values in your experiments, right? Okay, so in that case, you have to use the degree of freedom, because you use how many locus you use in your experiment, right? So if you want to do the know about the probability of using chi-square chart, you need to determine the degree of freedom, right? So degree of freedom means that the number of phenotypic possibility in your cross or in your experiment, and then you minus 1. That is equal to your degree of freedom. So for instance, if you have the Mendelian genotypes, which is the 3 possible genotypes, then your degree of freedom is equal to 2, right? So using this degree of freedom values, then you determine the probability of distribution using the chi-square table, right? Then you calculate that how much or how significant your observations is compared to chi-square distribution. So let's say if you have the degree of freedom 2, then you observe the values, you calculate the values around like 0.03, then this is something between these two. Then you can calculate that how much the chance your experiment is significant or not. Okay, so this is the examples. So let's look at the single locus, and you have the 3 possible genotypes as Mendel. So you have large AA, heterozygous, and small AA. And once you have the experiment, your expectation is 133, 59 to 6. That is followed by the Mendelian ratio. Then your observation is 108 and 71 and 1. And then you can easily calculate the differences between the observation and expectation, observation versus expectation, observation versus expectation. Then you calculate the numbers, and your chi-statistics is equal to 6.78, right? So given that chi-square statistics value, then you can back to here, then you see that the degree of freedom is 2, and the value is something between 5.99 and 9.21, because yours is 6 point something. Then you can say that your probability of the significance is around here, right? I mean, nowadays we can use the computation to calculate that number. Okay, so you can say that you reject the null hypothesis at significance level, which is the below than p-value lower than 0.05, right? So we can easily calculate that whether your observation is something, different from what the natural population normally have, right? So this case is significantly different and significantly, rejected the notion of the Mendelian ratio, right? Okay, so in other words, if you use the chi-squares, or if you use other, whatever the hypothesis testing, then if the null hypothesis is not rejected or is supported by the analysis, then your assumption is exactly same as the Hardy-Weinberg assumption. Or if not, then this is the different situation, right? So Hardy-Weinberg experiment is also used in Thomas Morgan's linkage experiment. So this is the well-known examples of why Mendelian second law has exception, because now we know that there is the phenomena called genetic linkage, right? So the hybrid fruit fly cross experiments done by Thomas Morgan at the time. So null hypothesis is that the 2 traits we used in here, black body eyeless fly and wild type. So we have 2 phenotypes here, black body and eyeless. These 2 traits are not linked to each other, because according to Mendel, it has to be asserted independently, right? and therefore the random segregation independently happening during the gamete formation, right? But once you do the F1 cross result, that the numbers is like this, and that one is once you put into your expectation, this is the different from than your expectation. Because if you do the D hybrid cross, which is if you use the two loci and your experiment according to the Mendelian ratio, then your ratio has to be 9:3:1 ratio, right? Because this is the 3:1 ratio times 3:1 ratio. That has to be 9:3:1 because two loci is totally independent to each other during the transmission. But in case your observation is quite different, right? So if you do the 10,000s of experiments, then your observations is compared to expectation has some of the deviation, right? So it's not exactly same numbers as you expected according to the Mendelian factors, right? So if you calculate that the observation numbers, then you put it into the chi-square statistics, then whether these two locus following the Mendelian rules or not, right? So this case, it has the linkage and it didn't follow the Mendelian ratio. It has something significant values in the chi-statistics. So we can say that it has something, going on in this loci other than the Mendelian factors. Now we know that this is the linkage factors, right? Okay. So calculating the chi-squares is about whether we describe the situation as we define in a natural population or not, right? So most of the the natural phenomena we see in an experiment is we can put it into some distributions developed by mathematicians in early times, like central limit theorem, normal distribution, binomial distribution, or whatever the distributions. But like chi-square statistics also, we put it into the chi-square distribution because we have certain frequency we can define by Mendelian factors. And then we observed our experiment and compared the numbers with the Mendelian expectation, then see how much that deviation from the Mendelian definition, right? Then we can use the chi-square and calculate whether this is the significance or not, right? So the things you have to know about that, we use the predefined distribution in this chi-square and Hardy-Weinberg the equations, right? And chi-square is working on the situation, and whatever the calculation or computation, we don't worry about that anymore because we don't have to do that the hand calculation anymore because now we can all put it into computation, right? So chi-square test is you can find all over the internet, and you can easily put the whatever the chi statistics. Okay, so we learned a few things during the Hardy-Weinberg topics. So first one is the Mendelian inheritance and segregant is independent to each other. And that is the huge implication from Hardy-Weinberg. And also the dominance defined by the Mendel is actually the molecular phenotype. And what that means, we're going to learn more detail in a later section. And then the Hardy-Weinberg equation, let's drive that by yourself. It's easy. It's the middle school equations. Don't be panicked by the lots of the mathematical notations on here, and then also the assumption of the Hardy-Weinberg equilibrium, because this is the sort of the link between the Mendelian factors to the population genetics, and also the latest biomedical genetics as well. And also think about the Hardy-Weinberg correlation, and also think about why we use the chi-square statistics in this situation, right? So that's all. And also one thing I can expect from the questions is probably people ask lots of things about what is the null hypothesis, what is the hypothetical testing, blah, blah, blah. And I don't know whether, I mean, null hypothesis or hypothetical testing is like, you know, this is like the basic units as DNA in a biology major. I mean, you can search some the materials even like in Koreans or many good, you know, the introductory materials you can search from the internet. Please make your words using, you know, from your understanding and describe that one during your questions. That is the critical things, not just, you know, asking what is the null hypothesis, right? I mean, null hypothesis we can clearly define in here that the null hypothesis is that no substantial statistical deviation between the observed variables and expected variables. I mean, I couldn't technically, you know, improve this definition more than this sentence, right? So please make this one into your words and also put it into the, I mean, if you want to give a questions about that, you know, topic, then digest on your words and put it into your question. That would be, I mean, much more helpful for your learning. Okay, so we covered the issue about the Hardy-Weinberg equilibrium and also some further reading. So these two things is quite, I mean, amazing. So, I mean, I don't know who wrote this one, but yeah, this is the one Hardy-Weinberg principles. This explained by Nature Education. I mean, this is quite introductory stuff, and this is more in-depth. Read on the how Hardy-Weinberg principles has been developed over the century, and that is quite great review on that. Is written by Oliver Mayer. Okay, so hopefully you enjoyed that the materials, and we are going to the next session on the this Wednesday, chromosome and sex, and then we follow up by gene and heritability. But as I instruct earlier in this session, these two topics could be quite extremely difficult if you don't understand these four. So please study before next week and up to this part. And if you have any questions and or not clear things, please leave your questions on our Slack. And hope you will prepare your exams on the end of this month. Okay, so that's all, and good, and see you next time.